after class today. If you have any questions, please put them in the Q&A box. I'm the only person moderating this today, and I'm also the teacher of this class. So if you put them into the chat box, I might miss them. But if they're in the Q&A box, I'm more likely to see them. And if we have time at the end, more likely to answer those questions. So please do that. If you just have general chatter, like oftentimes we use the chat box to say where we're tuning in from. I'm here in Seminole County in Sanford, Florida. You're welcome to use the chat box for just general chatter or commentary. If at the end of the class you want to be unmuted to ask a question and we have time, you're welcome to use the raise hand button to raise your hand and ask to be unmuted. Today we are going to be learning about how to identify mushrooms. And hi, I'm Katie McCormick. I'm the residential horticulture agent here in Seminole County, Florida. And I'm part of the University of Florida Institute of Food and Agricultural Sciences, which is part of the land-grant university system created in 1862. And it's meant to extend the research and teaching of the University of Florida to the residents in our state. It is funded by local, state, and federal funding. And it includes just about anything that has to do with living a healthy, happy lifestyle. So we include family and consumer sciences, which used to be home ec. One of our agents today is actually teaching cheese making this afternoon, which I'm kind of sad to miss. Agriculture and how to start a small farm business or a large farm business or problem shooting on your farm. Horticulture, which is my position here. I teach residential horticulture, so anything about growing things in your yard. And 4-H, which is where we teach kids all of those things, as well as how to be happy, healthy citizens in our counties. Extension is found in all 50 states and all 67 counties in Florida. So no matter where you live in the United States, you are going to be able to find an extension office not too far away. And please use us. We are paid for by your taxes. So you've already paid for this service. Please use us. So housekeeping done a little bit quicker than I normally go, so hopefully we don't have too many people trickling in after this. We're going to cover what is a mushroom, what are the key characteristics of mushroom ID and what you need to be aware of when you're trying to look at a mushroom and figure out what characteristics you need to look at. What supplies do you need for mushroom identification? What resources should you use whenever you're trying to identify mushrooms? A quick slide with the references I used for this presentation, and then I'll take questions if we have time at the end. There's a lot to go through, and I'm going to try to not go too fast through it. But if you need clarification, again, please use that Q&A box, and I will clarify at the end of class. Before we get started, though, I did want to give a disclaimer. On your journey to mushroom identification mastery, any unidentified mushroom is deadly to consume. So if you don't know what that mushroom is, do not put it in your mouth. No edible mushroom is safe until you are 100% confident in your identification. So again, if you don't know for sure what that mushroom is, do not put it in your mouth. Mushroom poisonings are tough to track reliably in our healthcare system, but some scientists estimate that they cause between around 10,000 illnesses a year and 100 deaths a year globally. Mushroom toxins can lead to extreme gastrointestinal distress, hospitalization, organ failure, and death. So please don't eat mushrooms unless you know exactly what they are. And that's part of the reason I'm not going to show you any slides of edible mushrooms versus deadly mushrooms today. I don't want you getting them mixed up. Instead, I'm going to give you the tools today of how you identify them yourself and figure out if they are safe or not. And it is on you to be 100% sure that that mushroom is safe. To highlight that, on the screen here, I have 3 separate mushrooms. One of them is considered a pretty choice edible, and the other 2 will kill you. They will cause organ failure, you will need an organ donor, or you will die. Can you see what characteristics would distinguish them from each other? So I know what characteristics I'm looking for here to determine which of these is not going to kill me, but if you aren't sure, you should not be trying to eat these mushrooms. And I'm coming at this class and our mushroom hike on Saturday from a standpoint of mushrooms are super fascinating. Knowing more about them just intrinsically will make your life a little bit better because you have something in the natural world that's wonderful to look at. Not necessarily we're identifying these in order to eat them. So with that dire threat and warning in mind, let's go into what is a mushroom. So fungi include the mushrooms that we're going to be talking about a lot today. They also can include molds, truffles, and yeast. So all of these are covered in that fungal kingdom. And of course, we're usually more interested in the mushroom and maybe the truffle side of things. Although I know a lot of people that like yeast between beverages they can drink and bread they can eat. If we look at the eukaryotic tree of life, so where everything is connected when we're looking at our DNA and our evolution, fungi and animals are pretty close relatives. And this is one of the reasons that when you have a fungal infection, it can be really difficult to treat for it. And a lot of times the treatments are right on the edge of what's going to hurt you as well. Because a lot of the things that react to humans also react to fungus. A lot of the things that we use to treat for fungus can also really harm humans because we're so closely related when it comes to how our cells are formed. Fungi are also heterotrophs. This just means they have to get their energy from other organisms. So they have to consume other things in order to produce more of their cells and to continue to live and thrive. Those things can be living plants. We have a lot of plant pathogens, particularly here in humid Florida, that attack our plants while they're still living. My favorite on this particular slide is the Ganoderma butt rot, which is the fungus conch growing off of the palm tree here, and will attack healthy palm trees. So that's a big issue here in Central Florida, and I've seen it quite frequently. Kind of fascinating to look at is the rust fungus here on the clover, which the rust will attack a lot of different types of plants depending on the species of rust, but really interesting to look at with that orange fuzz. The ones that we typically see out in nature and are particularly interesting to us would be saprobes, or the fungus that feed on dead plants and other decaying items. And you can see we've got some really interesting ones here. And they also form symbiotic relationships to get their food. So you see that with the mycorrhizal connections that fungi will make with plants in order to exchange nutrients for sugars with the plant. And you also see that with lichens, which are a combination of algae or cyanobacteria and fungus that work together to photosynthesize and then also break down nutrients on whatever surfaces they find themselves on. So you have a lot of variability with what fungi will eat, where you're going to find them, and what kind of habitats they will inhabit. They also have indeterminate growth. So theoretically, an individual could live forever as it just grows larger and larger and larger over time. Maybe some of these older connections break down, but you still have newer connections that are growing out away from the food that it's eating. So mushrooms can get quite big. And most of the mushroom is not necessarily, or most of the fungus is not necessarily what we think of when we think of mushrooms, because the mushroom is the top of this organism. Fungi use absorptive nutrition, so they're secreting enzymes and they digest the food outside of their bodies. So those enzymes are put onto the surface of whatever they are decaying, and then they take up the nutrients from that surface over time. And most fungi are multicellular. So their bodies are made up of microscopic filaments called hyphae. And the hyphae grow right at the tips. So they'll grow longer over time from the tips. And when they're all together, this is what we call the mycelium of the mushroom. So a bunch of hyphae together are mycelium. Some fungi are also unicellular though. So unicellular fungi without flagella are called yeasts. We won't be really covering those much today, but you probably interact with yeast pretty frequently in your life. Most fungi also reproduce by spores. So many fungi will make both sexual and asexual spores. Those spores get taken up a lot of times by the air and scattered around in the area that they're growing, and hopefully they fall on whatever their host is so they can continue to grow. And our mushrooms are just the sexual fruiting bodies that produce those spores. So here you can see Microcebe titans being held by Dr. Matt Smith, who is our mushroom identification specialist at University of Florida. If you've called poison control because of mushroom poisoning, they have probably sent your case on to Dr. Matt Smith for initial identification and information on what they should do. He is the mushroom expert for our state. Those fruiting structures, the mushrooms, are usually ephemeral. So they're only lasting for a short period of time. They are growing up, they're producing the spores, they're dropping the spores, and then they decay pretty quickly. There's no other reason for them other than that sexual dispersion of those spores. So you can consider the body of the organism when we're talking about mushrooms to be that mycelium that we talked about earlier, kind of like we've got our trees that have all those leaves and branches and twigs. That's what our mycelium is. And then the mushroom is the fruiting structure, just like an apple would be the fruiting structure of a tree. So the tree generally isn't going to hold on to that apple forever. It's briefly got it on there, it ripens it up, and then it's dispersing those seeds. That's the same thing that mushrooms are doing. They briefly appear because this is the fruiting structure. They are dispersing their spore, and then they disappear, and the mycelium remains on whatever the substrate is that this is growing on. So whether that's decaying wood or a living tree or an animal, this is where our fungus kind of differs in what we consider kind of growth habits. So outside of just what is a mushroom, if you are trying to identify a mushroom, what are the things you actually need to look at? And I feel like learning these key characteristics can start to get your mind working on seeing them. I know for plant and insect identification, as I start teaching people, like, this is what we're looking for, eventually the more you practice that, the better you're going to get at seeing these different features inside of a plant. But when you're just starting out, a plant or a bug or a mushroom, a lot of them look very similar. So hopefully this next part of the presentation really breaks down what you're going to be trying to look for whenever you're doing mushroom identification and gives you a good starting place for that. So if we're looking at a simplified view of the fungal tree of life, most of the mushrooms that you're going to be interested in are those classic mushrooms, and most other fleshy fungi are going to be in Basidiomycota. And this is a particular branch of mushrooms that produce their spores on microscopic pedestals called basidia, which you can see a picture of here. These are the spores. These are the basidia holding them. And you can see this one has 4 spores per basidia. And for the most part, this is all of our classic mushrooms, as well as several different truffle species. The other main part of the fungal tree of life that we typically are going to see are the cut fungi, true truffles, lichens, and morels. And these produce their spores inside microscopic sacs called asci. And these are Ascomycota. So most of the mushrooms we see are going to be in Basidiomycota. The rest of them are going to be in Ascomycota. And together, this makes up about 120,000 identified species worldwide. So you've got a lot of species between these two, but most of the mushrooms we're looking at in nature and are interested in are going to be in these two. Other fungi are about 10,000 identified species and make up some kind of weird stuff that you'll see occasionally. And that does include our yeasts and our molds. And a lot of those fungus that'll attack animals are actually in this other fungi, which you can see here. We've got an amphibian chytrid fungus that's causing disease on amphibians here. It actually eats the keratin out of their skin. So this is where we've got some of our creepier pathogenic fungi. If you want to be somebody that gets to name some scientific or provide a scientific name, probably going into mycology wouldn't be a bad bet. So for fungal species diversity, we have about 130,000 described species of fungus. And the estimated total diversity of fungus, if we're looking at all of the mushrooms worldwide and all of the other funguses, is probably between 1.5 and 5 million unidentified species. So again, if you want to be somebody who gets to name a species, studying mushrooms wouldn't be a bad bet. Going a little further into those key identification aspects, you're looking at the whole mushroom when you're trying to make an identification of it. So this is just parts of a mushroom drawn out for you to take a look at, kind of broken out. You can see we're looking at how do they grow when they're starting out. This one starts in a button stage. Does it have teeth or spines on the underside? Does it have a pore surface? What does the top of the cap look like? What does the stalk look like? What does the base of this mushroom look like? Getting even further into this, does it have a smell? Does it have an interesting texture if you squish it? Does it produce milky latex whenever you cut into it? Does it discolor a certain way when you cut into it? There are a lot of different things we look at when it comes to mushroom identification to determine what this mushroom is. So for our distinguishing characteristics, what does the cap look like, or the top of the mushroom? What are the gills shaped like? What do the spores look like, both color and if you have a microscope or access to a microscope? What is the shape of those spores is in a lot of our keys to mushroom identification. And then does it have a ring or skirt? And what is the shape of that ring or skirt? We also look at what is the shape of the stalk. Does it have a stalk? Is it just coming directly out of the ground or the substrate it's growing on? Where is the stalk connected to the cap? Does it have a veil, like this very interesting stinkhorn here? Does it have a vulva or cup at the base of it? And sometimes even what does the mycelia look like whenever it's growing out of the substrate? What color is it? How is it formed? So taking a little bit of a closer look at those features, let's start with our spore-bearing surface and spores. So our spore-bearing surface is usually under the cap of the mushroom, because when you're releasing spore, you're going to be releasing it down around you onto the substrate you're growing on. And it can come in a couple of different shapes for our mushrooms that do have this spore-bearing surface on their underside. So you can have gills, which are wide and thin and sheet-like that radiate from the stem. You can have pores, which are many small tubes ending on a spongy surface. You can have ridges, which are short, blunt, elevated lines on the stem and under the cap. You can have teeth, which are like many small finger-like projections sticking down from underneath that cap. When we look at those on actual mushrooms, you're going to see with our pores here, you can see it does have that kind of porous surface. So there's those little holes underneath the cap, and that's where those pores would produce the spores. You've got our gilled surface here with those nice ridges going throughout. We've got our tooth surface here. You can see, as opposed to the pored surface, which is kind of flattened and then has holes in it, the tooth surface has these little projections coming out with the space in between them. Down here, we've got a smooth or ridged mushroom. So you can see you've got a little bit of texture here, but it doesn't have gills like this one up here. And over here, we've kind of got a combination of gills and ridges and maybe some other features going on. So not every mushroom is going to fall within these four. When we're looking at the gills on gilled mushrooms, you might also consider the spacing. So they can be very crowded, like you see here on this mushroom. They can be close together, or they can be distant from each other, like on this mushroom. So pore spacing can also be one of our key identification characteristics. Spore color can also be a very key characteristic. So remember, our fungi are reproducing by spores, and that spore color can really tell you a lot about what species you're working with. Spore prints are a powdery spore deposit visible to the naked eye, so it's pretty easy to make these and usually pretty easy to see them as long as you have a good background. And sometimes nature will make the spore print for you, which we'll look at in just a second. If you want to make a spore print, what you need to do is cut the stem off of the mushroom, and you want a not completely like decaying matured mushroom, but you want a mature mushroom when you're doing this for best spore print. You want to cut the cap off, put it on a sheet of paper or aluminum foil. The paper should be either white or black. Aluminum foil, I find, makes it a little easier to see if you have white spores or if you have black spores. There are some mushroom guides that will actually tell you to use a piece of paper that's white and black down the middle, and you stick your mushroom right in the center of that, so you have spore dropping on black and white. You're going to leave that mushroom underneath the cup on the surface of whatever you're collecting the spore print on for about 24 hours. Sometimes you can get a spore print as little as 2 hours, but 24 will give you the best result. And then you'll pull that mushroom up and you'll get your spore print from it if you have it spore side down for one, and if it hasn't already dropped all its spores for the other. So this one made a really nice dark spore print. They must have picked it at just the right time. When we're looking at spore prints, they can come in a lot of different colors. So here you can see some pink spores, and this is where the mushroom has done the spore printing for you out in nature. So it dropped these spores on the white top of the mushroom beneath it, so you can see them pretty nicely. This one was done out in the field where they did it on a piece of wood. So, I mean, I guess you have some color contrast there, but not the most recommended surface. But you can see these are really nice pink, kind of mauve spores there as well. Here out in nature, we've got these nice white spores growing on this particular mushroom underneath the one that's dropping them. And on this one, you can see our white spores, kind of like snow at the base of it on the leaves. So you get a lot of interesting spore colors. One to keep in mind, I guess, for Florida, if you're trying to do identification, is if it has green spores, it is probably Chlorophyllum molybdites, which is commonly known as the vomiter, although there are a lot of mushrooms commonly known as the vomiter. This one is the most frequently eaten poisonous mushroom in North America, though. So it'll produce these really interesting kind of teal green spores. And if you do accidentally consume this one, you're in for a pretty bad time. You'll have gastrointestinal distress, including vomiting, diarrhea, and colic, often severe and occurring 1 to 3 hours after you eat the mushroom. So far, nobody's died from the gastrointestinal distress this causes, but you will be having a very painful stomach for a pretty long period of time if you eat this one, which is just another caution: don't eat these mushrooms if you don't know what they are. But again, one sure sign you have this one is the green spore print it produces. Keep in mind, I guess it kind of showed on the previous slides, but remember the spores are not always the same color as the mushroom. So this beautiful red bolete here has brown spores. This beautiful orange mushroom here has white spores. This really pretty little purple mushroom here has white spores. And this interesting white or purple mushroom here has rusty brown spores, almost trending toward orange. So just because the mushroom is a certain color does not mean its spore print is going to be that color. Outside of spores, we get into looking at how the mushroom itself is really formed. So both where the gills are attached, if it has gills, and what is the shape of the cap if it has a cap. And you can see we've got a lot of different ways that the gills can be attached to the mushroom and a lot of different cap morphologies. When I send you guys the video link at the end of class today, I will also be sending you some different identification resources, and this particular slide will be among them. So taking a closer look at a couple of these, you can see we've got our free gill formation here where the gills are not attached to the stem. And you can see we've got this ring of gills, but they are well clear of where this stem attaches to the cap in this image. You can also have decurrent gills. So these are where the gills run down the stem for some length. And you can see these go down that stem from the cap for a decent amount of space on this mushroom. They're taking up quite a bit of the stem. If we're looking at cap morphology, you've got, again, a couple of different caps you can see. And what I'm showing you is not all of them. That would take more than an hour. But I wanted to just highlight some of the differences you might see and get your mind thinking about what you might have seen in the past. So we can have conical or triangular caps, which is like this little guy here, kind of looks like a hat that a fairy might wear. You can have umbilicate caps, which will have a small deep depression. And umbilicate is just like umbilical cord or like your belly button. So you can consider umbilicate like a belly button. I've seen some that are hairy and it's kind of disconcerting how much like a hairy belly button they look. And another interesting one, in my opinion, is umbinate, which is where you have this central little bump or knob right at the top of the cap, which you can see here on the screen. The surface of the cap can also have different textures. So they can be completely smooth, kind of like what we see here on this mushroom. They can also be velvety in texture. They can be hairy or fibrous, kind of like that mushroom that looks a little bit like a belly button I talked about earlier. They can have raised scales, they can have flat scales, and they can have patches. So on the images here, we've got a mushroom that has some patches on it. And on this side, this is actually a hairy or fibrous mushroom with that umbilicate cap on it. So you've got that kind of fuzzy belly button thing going on with this one. Other things to look for on the mushroom is the cap surface viscid, or is it sticky and tacky when moist, or is it slimy or glutinous, or is it dry? So that texture on the top of it can really help with identification as well, and trying to narrow down what species you're looking at out of the 120,000 found around the world. And also, what does the margin of the cap look like, or what does the bottom of the cap look like? So it can be smooth or entire, crenate or scalloped. You can be striate with little cuts into the base. It can be wavy. It can be appendiculate, or with little appendages like tentacles on an octopus or antennae on an insect. Or it can be rimose and cracked, where you have this kind of cut-in look on the base of it. So again, just looking at a couple of those, you've got the cap margin on this one, which is crenate or scalloped. Really pretty texture, right? And you can see how this kind of looks like a scallop. You've also got striate margins here. So here again, we've got those kind of little almost gilled top to this mushroom. And this one, the appendiculate part of it hasn't grown out quite as far, but this would, if it was allowed to mature a little further, have even longer appendages than it does now with that fuzzy looking cap. And then getting toward the end of our fungal identification pictures, at least, what do the stem look like and what does the ring look like? And this is part of the reason why when you're doing mushroom ID, it's really suggested that you try to get the base of the mushroom if possible, because sometimes that base, which can be just straight down into the soil and then the mycelia started right at the base of it, or rounded or bulbous, or it might have that volval cup. Sometimes they'll have a rooting, kind of like a taproot you'd see on a carrot, or with much thicker rhizoids as opposed to just immediately turning into mycelium. And all of those are identifying characteristics, and particularly if it has a cup or not, can really distinguish between a lot of our super poisonous mushrooms and those that are edible, because oftentimes if it has a cup, it is going to be in one of the genuses that is containing a lot of those poisonous mushrooms. Also, the ring shapes can tell you a lot. So is it hanging down along the stem? Is it flaring out like a little skirt? Is it sheathing or pointing upward? Do you have two sheaths? Do you have cobwebs coming from the base onto the stem? Do you just have a ring zone where maybe a ring was at one point and no longer? All of those can be identification characteristics as well. So if we're looking at our sheathing here, you can see up here, we've got a mushroom with a nice ring on it. And if we're looking at our guide, this is probably going to be considered a flaring ring, because you can see it's kind of spread out from that stem. Down here, this one's very obviously our cobwebby ring, because again, you've got kind of that cobwebby look to it. For our bases, you can see here, we're dealing with a mushroom that is probably going to be considered bulbous, because we have that nice hip to it on these bases here. With this one down here, we're dealing with a rooting mushroom. It has that root that's coming down from the stem beneath the level of the soil. Other things you should consider when you're doing identification are context. If you are handling this mushroom, is it hollow or solid? If you pull apart the flesh, does it pull away from itself cleanly, or is it more like string cheese when you pull it apart? What color is the flesh of the mushroom? Does that color change if you cut into it or scratch it? Is there any interesting texture to it? So is the internal flesh, is it textured? Is there any other interesting texture on the outside of it? When you do cut into it, does it produce latex or a milky substance or sap? Some mushrooms just in nature will also produce guttation, and that's where they're actually producing liquid off of their caps just as part of their sexual reproductive strategy. So you'll see them out in nature and they might have a liquid just sitting on the cap in little pores of or droplets of this liquid. Really disconcerting. There's a mushroom that's a white mushroom and it produces a red liquid that sits on its outsides in little globules, kind of looks like drops of blood. Also, how do they grow? So are they gregarious or growing in loose groups where you see them kind of grouped together but not in a dense mat? Are they cespitous, which is where you see them in that really dense cluster? Or are they growing solitary one at a time, pretty spaced apart? And then really, really important if you do decide to get into trying to figure out edible mushrooms is what substrate are they growing on? Because oftentimes that's going to be one of your defining characteristics for, is this an edible? Is it edible on this substrate, or is this going to be poisonous? And that'll be, is it growing out of the ground off of something decaying under the ground? Is it growing directly on wood? Is the wood living? What kind of tree wood is it? So is this a maple tree, an oak tree, a pine tree that it's growing off of? Are you finding it in a field or a forest? Is it growing off of dung, or is it growing off of decaying leaves? What exactly is this mushroom eating? Other things to keep in mind is, what is the ecosystem you're walking through? Are the nearby trees hardwoods, or are they conifers? And then also your geographic location and time of year. So with our climate shifting and changing, sometimes the time of year can shift and change for some of these species, but they do have a general time of year and climate that they want to be growing in. And typically they're going to have specific geographic locations they're found. That mushroom on our first warning slide that I had earlier, where I said, look at these 3 mushrooms. If you can't identify them, you'll probably die. One of those has a extremely edible look-alike in Asia, and it's often used in soups and stews. And most of the people that are really harmed by poisoning from that mushroom misidentify it as the extremely edible, tasty one from their homeland and make a soup or stew out of it. And that's definitely enough of that mushroom to kill you. So it's a really good idea to consider, is this mushroom going to be available as an edible where I am right now? And other things to keep in mind is how big it is, the shape, which we talked a lot about already, the taste, which if you are tasting mushrooms, this is one of those things where you touch it to your tongue to see if it has a flavor, and then you spit that out and maybe rinse your mouth out and spit it again. You're just getting a little bit of that taste in your mouth to see if it has a taste. I would do that last if you're going to be using it for identification purposes. And does it have an odor? And when it comes to odor, there have been several different research papers published on odor, one of which is available as a free to download paper if you want to look at this chart a little bit closer or read more about it. So this one was a review on edible mushrooms as a source of special flavoring. And then they categorized the odors of these mushrooms and created these really interesting charts, breaking down our edible species into what they smell like. So you can see they can have a fruit odor, floral odors, they can have a rancid odor or fat and roasted odor, mushroom or earthy odors, vegetable odors, acidic odors, caramel odors, and there are several different species in that one, or sulfurous odors, all of which can be included in some of these edible mushroom species, and which, you know, if you've got a good nose, you could probably sniff out. There was another paper published in the journal Fungi about the distinct odors of mushrooms, and they just created an odor wheel to categorize those distinct odors. So they broke them down into chemical odors, vegetable odors, herb and spice odors, floral or tree odors, sweet, fruity, nutty, and animalic. And then they broke those even further down into different things that they'll smell like within those categories. So for instance, under floral and tree, you've got cedar as one of the smells that you might get off of a mushroom. Under animalic, I think my favorite here is old gym socks, although Boolean cubes is kind of fun too. So both of these charts can be useful whenever you are sniffing out a mushroom and trying to categorize what exactly does this smell like, because they really break down the most common scents you're going to get off of smelly mushrooms. Next, I'm going to, this isn't going to be a very long slide, but what supplies do you need for Mushroom ID? So you can start out Mushroom ID pretty easily with just buying one of the resources I'm going to be talking about earlier or in a minute for identification, and then going out and taking pictures and maybe collecting some mushrooms and doing some spore printing. But if you want to make up a nice semi-professional kit to do some mushroom hunting, having a nice pocket knife in order to try and cut the bases of these mushrooms either off of wood or out of the soil can be really useful. Containers to keep the mushrooms in, and in particular, again, if you're doing this for ID purposes, a tackle box with all those little tiny containers for you to stick your individual mushrooms in is really useful. And that's what we'll be using on our hike this weekend. Or small lidded containers, things like lidded yogurt cups, or one of our staff members here really likes the Talenti gelato, so we have a whole bunch of gelato containers with those screw caps that we use. Can be really good for larger specimens. Foil or paper for spore prints can come in very handy if you want to do spore printing out in the field. Maybe you're going on a longer hike. Also, for really large specimens, this foil can be used to wrap your very large specimen, like that Titan mushroom we saw earlier in the presentation, in order to bring it back and keep it from dropping spores or desiccating before you get a chance to look at it closer. Sharpie pen or pencil in order to label your finds, a basket bag or backpack to keep everything in, a notebook in order to record field data on where you collected the mushroom, what it was growing on, just to help you keep in mind what one it is. A note or insect repellent, because we live in the state of Florida and we have a lot of mosquitoes that would like to drink your blood while you're out mushroom hunting. Binoculars, just in case you want to look at mushrooms up higher in a tree that you can't quite reach. Magnifying glass in order to look closer at those identifying characteristics of the mushroom. A phone or camera so you can take pictures of where you see that mushroom in the landscape or in the ecosystem. And water, because it is Florida, you're probably going to be a little bit sweaty and needing to rehydrate at some point. So again, if you just want a starter kit, your phone, maybe something to write on and with, and a knife would probably be fine. I would definitely recommend the bug spray and the water to go along with that. If you want to do the full kit, this is what I would recommend for some serious mushroom hunting. The other thing you should have is some kind of field guide to mushrooms in your area. So what resources should you use? Our region has very distinct fungi, and many guides to North American fungi aren't really going to cover Florida and the Gulf Coast, because our climate is very different from a lot of the United States. So if you're looking for some good mushroom guides, Mushrooms of the Gulf Coast States is a nice one for our area. Mushrooms of the Southeast covers quite a few mushrooms you'll find here in Florida. The University of Florida Press has one of the cheaper mushroom books you can find for Florida, which is Common Florida Mushrooms by James Kimbrough. But the pictures in this are pretty poor. I don't know if it was a print problem or if they were just very blurry pictures when they were taken. I wouldn't say this is the best one, but if you're just getting started, it's not a bad one to have. And if you really, really want to get into mushrooms and know everything about their identification and be able to key them out really easily, Mushrooms Demystified by David Aurora is awesome. It is like an encyclopedia, though. It's about 2 to 3 inches thick. Doesn't really have a lot of pictures. It's almost all dichotomous keys, which is kind of like a choose your own adventure for scientists, where you see your first 2 sentences where it might say it's a gilled mushroom, it has no gills, and then you pick the number that corresponds to your identifying feature and continue on selecting for those identifying features until you finally get to the species that the book thinks you're looking at. So again, if you really feel like becoming a mushroom scientist, Mushrooms Demystified would be an awesome book to get started with. I just ordered one off of Amazon, so I'd have 2 for the hike on Saturday, and it's about $26, so it's not that expensive to purchase either when it comes to a huge scientific book like that. If you would like to avoid buying books, you can go to a couple of different online resources. So mushroomexpert.com, which started out as a website by a English teacher in, I think, Ohio, is one of the really good mushroom identification websites out there now. So you have a bunch of different mushrooms. He has a bunch of different mushroom identification keys you can use, and a lot of really great images and articles on those mushrooms. A lot of people contribute to this particular website now, and I know it looks a little bit like it's from the 1990s or early 2000s, but it is an awesome resource. Other resources that you can use are mushroomobserver.org, iNaturalist.org if you're wanting help with identification. That one has a lot of both amateur and professional identification people on it that'll help with ID if you upload pictures and your location. And there are also Facebook groups such as Florida Mushroom and Fungus ID that you are welcome to join, and they will help you with your identification. I accidentally ate a mushroom and I don't know what it is, which after this presentation, I hope you will never eat a mushroom and not know what it is standpoint. But in the case of accidental possible poisonings or pet poisonings, you can reach out to our mushroom expert at the University of Florida, Dr. Matt Smith, or Poison Control, and they'll reach out to him and they will help to identify those mushrooms. But for just I found a cool mushroom in the landscape, iNaturalist.org is really awesome, or learning to identify it on your own is really great. Florida Mushroom and Fungus ID also is a great site to use for this is an interesting mushroom I found posts. And the reason I distinguish those two is Dr. Matt Smith is identifying mushrooms to make sure people aren't dying. And if you're just sending him a huge bunch of pictures of cool mushrooms you found in your landscape, he is going to be bogged down by those instead of helping the people that need it. So just keep that in mind. So from the presentation today, the references and many of the pictures in this presentation were taken from 3 separate identification presentations presented by different people at University of Florida. So Fungi and Mushrooms 101 by Dr. Matt Smith was a lot of this presentation. Sarah Prentice, who is in the mycology department at UF, also provided a lot of the really cool pictures of the spore prints, and mushroom biology and identification presentation by Armando Ubeda, who is a natural resources agent in, I want to say, Sarasota, also provided some pretty good pictures in this presentation. And with that, I'm happy to take any questions. For this Saturday, the hike is sold out, so I don't have any more space in that one. I do plan on offering some more mushroom hikes in the future. I just need to figure out when I'm going to fit them in. When I do determine when those hikes will be, I will be posting them on our Eventbrite. Our Eventbrite page is something that you can follow. You can see the link on the screen there, or you can use the QR code to access that particular website. I'm also available to be emailed at k.mccormick@ufl.edu, and the Master Gardeners are available at the Master Gardener at seminolecountyfl.gov. So I got the question, what is the most common edible mushroom in our area? We do have chanterelles in this area. We have some oysters, although oftentimes the oysters are so buggy, you don't want to eat them. Occasionally you'll see lion's mane. There are also some that are less choice but edible, that I wouldn't necessarily like seek out, but if you find them, they're okay to eat. And one of those is that hairy looking belly button mushroom I talked about earlier. It's pretty woody, but it won't make you sick to eat it. How many deadly mushrooms are there in the world? So deadly mushrooms make up about 1% of the mushrooms worldwide. So when we're looking at the world as a whole, of the mushrooms you've got, about 1% will cause organ failure and death. Most of them, about 20 to 25%, depending on where you are going to cause just gastrointestinal distress. So you're going to be vomiting and have a very bad stomach and maybe some blood coming out of both ends for a couple hours. About 20 to 24% are edible but unpalatable, and then something like 10 to 20% are edible and palatable. Should you use protective gloves when handling mushrooms for identification? So our mushrooms have to be ingested in order to make you sick for the most part. I'm somebody who has really, really sensitive skin, so I wouldn't be surprised if there are some mushrooms out there that maybe will cause a reaction with my skin. Like I can't even wear most of our sunscreens. I have to go for like the zinc only. So when you see me on the hike on Saturday, my face will be white. But for the most part, for most people, just handling the mushrooms, as long as you don't then like lick your hands, should be fine. And even for our really deadly mushrooms, most of the time you have to eat like a full mushroom in order to get sick off of those. It's not just like a little bit. So in the chat, which again, put your questions in the Q&A box if possible, because I might miss them in the chat. So what was the name of the Southeast specific books I referenced? Those are Mushrooms of the Gulf Coast States and Mushrooms of the Southeast. I will say for keys, I feel like both of these start at the like microscopic looking at the shape of the spores level, which isn't always super helpful. Some of them do start at like what's the shape of the mushroom, which is a little bit more helpful. I like guides that start with what's the spore print color. I feel like that's a really nice determining. And then how do mushrooms and fungi differ from slime molds in terms of reproduction? You know, I believe slime molds also produce spores, but I'm not 100% certain on that. I've been focusing mostly on mushrooms for mushroom identification purposes. Will there be any hikes in North Central Florida, Gainesville area? Oh, so if you are in North Central Florida or the Gainesville area, there actually is a group through the University of Florida that you can join, and you are welcome to join. I didn't include them in here because I wasn't sure how many out of county people we'd have, and I didn't want to send people their way. The group name is Falafel, and I cannot remember what the acronym stands for now, but if you look up Falafel Mushrooms Gainesville, I think you'll find their Facebook page. I was looking just yesterday and they had 3 hikes over the last month. So they're going out almost every weekend for a mushroom hike. How does the slime work in the reproductive process? You know, I'm not super sure on that. Whoever's super interested in slime molds, if you email me that question, though, I'm happy to research it for you. Are there cases of allelopathy? Are you talking about like in the natural world? So I mean, I don't believe that mushrooms are generally going to be interacting in a way that keeps plants from growing, because oftentimes they need them. They do work with their mycorrhizae to help plants to grow for some species. But again, with 120,000 species of mushrooms, there's probably one that is going to do something against plants growing. And some of them are plant pathogens. So you do see them colonizing living plant tissue and breaking that down. And we see that with a lot of our plant diseases. Those will be pathogenic fungi. All right. Then thank you for joining me today. I can now see that I can add a little bit more information to this because I got through the information before, so maybe I'll add some more pictures of caps and stuff. Will you be providing a link if we want to view this again? Yeah, so after class today, I'll be sending out an email with a link to this recording once I get it up on YouTube. And I'll also be compiling some different resources that you can use whenever you're trying to identify mushrooms. So I'll give you the list of books, as well as attach a couple of the different keys you can use for fungal identification. Can I talk about cordyceps and other zombie fungus? So cordyceps are a pretty interesting fungus. And the way they work is the spores will come out of an insect or other organism that they've colonized. And usually that insect or other organism will crawl up above whatever its species is where they're nesting, and the spore will rain down on their friends and relatives. And then those spores will be taken up by the insects, either breathed in or ingested, and begin to grow inside their bodies. And generally, they'll colonize the nervous system and brain of that insect over time, forcing it to behave erratically and against its usual instincts. Eventually, that insect will also be encouraged to climb up to a high place and rest. And then the fruiting body of the cordyceps fungus comes out of them and again rains down the spores on their friends, starting that process all over again. There was a University of Florida entomologist or entomology student recently that found it's not only happening with ants, which is where we commonly see that because they're such a social insect group, but she actually found it in some wasp species in the forests around Gainesville. What is the blue mushroom on the cover of the book? You know, I'd have to look it up. I believe that is one of the blue mushrooms that produces the milky latex sap, but I'm not 100% sure on that. I'd have to look it up in the book. What is the deadliest mushroom? We have two in our area for sure that are deadly. I was just looking up some of them today. So the two that are super deadly in our area that actually were in, let me go back to the beginning of this presentation on my warning slide, but the two super deadly ones that you can find in Florida, and one of them was part of a poisoning recently where two men ended up in the hospital after making a stew out of them. Let's see. Almost there. Went past it. Is the Destroying Angel and the Death Cap are the two common names. Both of them are in the genus Amanita, which Amanita contains a lot of the mushrooms that'll make you ill. These two will cause organ failure though. So I guess spoiler for those that hung out till the end, we've got our Destroying Angel up here at the top, or Amanita bisporigiera. And then we have Amanita phalloides down here at the bottom. And the choice edible in this image is Acaricus campestris. So we've got Destroying Angel for our common name, Death Cap for our common name, and then common meadow mushroom for our common name on this slide. And I hope after this presentation you can see some of the features you might look at to try and distinguish between these three. In particular, though, amanitas usually have the volval cup down here at the base, and they'll often have a flared ring around the stem. So both of these are found in Florida. I was looking on iNaturalist, both have been reported in our area of Florida as well. So again, like, I don't want to tell you guys not to eat mushrooms that you find, but you need to be 100% sure the mushrooms you found are not going to kill you or make you really sick. I don't want anybody getting sick. All right, I'll wait another minute or so for any other questions. And if we don't have them, I'll go ahead and end for today. Can you be arrested for possessing the hallucinogenic mushrooms if you collected it for ID and didn't know? You know, I'm not sure on that one. I would have to ask our sheriff. I would say if you're not holding a whole bunch of them, I don't know that you'd get in trouble for that, but I don't want to be held to that. So I'm not sure on that one. I think it'll be up to whatever cop finds you. And I guess how harmless you seem with all of your other mushrooms. But, you know, if you're finding mushrooms growing off of cow patties, maybe don't collect those ones. All right. And thank you all for joining me. For those of you coming on the hike this weekend, I look forward to seeing you. And I hope for those of you that can't come on the hike this weekend, that maybe I'll see you in a future hike. With that, I'm going to go ahead and end for today. So yeah, thanks for joining me and have a good afternoon. Good luck with your mushroom hunts.